All right. So I want to thank those of you joining us in person and virtually. We've got a nice list of people. This is the first CTSI seminar for the regenerative medicine theme this academic year, and I'm very excited to get us started. So our speaker today is Dr. Hanna Mikkola, who got her MD and PhD in Finland. She did postdoctoral training at the Lund University in Sweden and then at Harvard Medical School with Stu Orkin. She was then recruited to UCLA to join the Department of Molecular Cell and Developmental Biology, as well as to be part of the Broad Stem Cell Research Center. So she was a big recruit for us. She's been heavily involved in a number of organizations, the ISSCR, ISEH, ASH, including serving as president of ISEH. She's been awarded a lot of important honors, a V Scholar, Leukemia Lymphoma Society Scholar, McCullough and Till Award, Rose Hill Scholar, a number of innovation awards. And she's made just tremendous contribution to our understanding of the development of stem cells and progenitor cells in the blood system, particularly in the embryo. Her group has really helped us understand where blood stem cells develop, how we can isolate and identify them, how they are maintained in terms of their self-renewal capacity. And most recently, her group has mapped at single cell resolution the development of blood stem cells from their emergence from hemogenic endothelium all the way to birth, which is just a really remarkable discovery and contribution to the field. I've known Dr. Mikkola since around, I think, 2006. She was a great member of my thesis committee when I was a graduate student. And so to see the way her research has evolved over that time, filling in the gaps of the models that they showed back then, and now understanding these important transcriptional regulators and markers to be able to follow this process. And she's had this singular goal of trying to be able to make and expand blood stem cells in the lab for the purpose of transplantation for patients. And it's such an incredible representation of fundamental biology with the goal of translational clinical purposes. And so I'm very excited to have Dr. Hanna Mikkola give our first regenerative medicine theme CTSI talk today. So thank you.
Okay, now the sharing part. Okay, thank you so much, Andrew, for the invitation and incredibly thoughtful introduction. And thank you all for coming, and thank you all for those who are sitting in your cozy home offices. So yes, today I would like to go talk about this one question that I've had since I came to UCLA 17 years ago, and that is how to make a human hematopoietic stem cell. So let's see this. We're not moving.
See if that.
Okay.
Now see if it works.
No, all right. So we all know the implications of hematopoietic stem cells in therapies. We can use them to permanently cure many devastating blood diseases such as leukemias and inherited blood disorders. However, the challenges that have been there since I started working with hematopoietic stem cells already in '98 in Lund, the fact that we cannot really robustly expand hematopoietic stem cells in culture, and then that it still hasn't been possible to generate fully functional hematopoietic stem cells from pluripotent stem cells. In '98, this was the human ES cells that were just arrived, and then more lately, the iPS cells, which could now serve as an unlimited, personalized source of hematopoietic stem cells. So all these wonderful technologies such as gene editing and they are hindered and held back because we don't know how to maintain or make hematopoietic stem cells in culture robustly. And really comes down to the question that we don't understand about the fundamental mechanisms, how human hematopoietic stem cell stemness is established or maintained. So in every day, the stem cells need to be making decisions whether to stay sleeping, which is what they do very well, or whether they start self-renewing either symmetrically or asymmetrically, depending if you need to expand the pool or just maintain homeostasis, or whether locate to a different location or start differentiating. When we place HSCs in culture, they stop sleeping, but they also don't know how to self-renew. Everything is faded towards differentiation or apoptosis. But what's really happening in culture hasn't largely not been understood. If we think about the prospect of making a hematopoietic stem cell and wondering how should we do it, and you want to ask the expert, the embryo, how to do it, the embryo says it's very complicated, that it takes a long time, and we need many different environments, because we know that hematopoiesis evolves during development in many different anatomical sites and locations that really give something unique to the developing hematopoietic stem cells. And what's also complicates this is that before HSCs are formed, there are a number of waves of hematopoietic progenitors that don't come from the HSCs, but are generated independently. So when you're starting to study the HSC, you don't even often know what cell you are studying. So if you can't find the cells, if you don't know in which location they are, and how to distinguish the self-renewing or prospectively self-renewing hematopoietic stem cell from the progenitors, how are you going to make progress? And without this, we can't really know where exactly are we failing in our pluripotent cell-derived cultures. So over the years, we'll learn about several important regulators. Now we know how to break the system. So from SCL knockout studies told us that SCL is critical to specify the hematopoietic lineage. And not only that, but it's also suppressing an alternative mesodermal fate, such as the cardiac fate. And SCL turns many important downstream hematopoietic transcription factors, such as RUNX1, that is the key factor that turns on hemogenic fate in the hemogenic endothelium. We've now learned that HSCs emerge from the dorsal aorta on the ventral wall from this unique type of hemogenic endothelium. We know it's there, but we didn't know exactly how it looks. After that, but the SCL and RUNX1 don't decide whether to make an HSC or progenitor. So from our failures to try to make a self-renewing HSC from human ES cells, we learned that the inability to activate HOXA genes, which was because there was not enough retinoic acid signaling in these embryos, this is why we couldn't make the right type of cell. So now we learned from our failures that the retinoic acid signaling and HOXA genes are absolutely critical for getting them on the HSC fate rather than to the transient fate. But we still didn't know how to make them self-renewing. So we did better markers for the HSCs. We found a surface marker GPI80 that enabled us to identify the self-renewing HSCs from fetal liver. And from this, we could really have a deeper look into the transcriptome of the HSC. And from the studies where we looked at every time when we lose self-renewal, what are the factors that always go with the self-renewing cell? We learned about new mechanisms of self-renewal. And we identified this factor, MLOT3, AF9, that we then learned is a critical for maintaining and expanding human hematopoietic stem cells. I'll come back to this a little bit later. But there were still many gaps in kind of our understanding of this full. How do we really connect these dots to each other? And we really had to wait for the technologies to develop to do that. So the key questions we wanted to ask is, how do you identify the newly emerged nascent HSCs compared to these progenitors? Because neither of them is robustly transplantable at this point. What locations support HSC development? We know that they're made here in the AGM, they go to the liver, but we and others have shown hematopoietic stem cell activity also in these extraembryonic tissues. How do they play into this? And then, very important, what is the process that the nascent HSCs have to kind of execute in order to do their ultimate job, to be a self-renewing graftable hematopoietic stem cell? We call this functional maturation, but we didn't really understand what it means. And then finally, what makes this HSC forming hemogenic endothelium unique and different from those other types of hemogenic endothelium that doesn't make an HSC? So as I said, we really had to wait a couple of decades for these technologies to develop. And this is where the single cell RNA-seq really came to rescue. So we worked with the Katja Schenkel-Lehlen lab in Germany, and they were able to give us access to these very early human embryos, exactly the time when HSCs are emerging. So we profiled starting from the 4 to 5 week human AGM, placentals, yolk sac, fetal livers at different stages, and compared to the neonatal HSCs. First, zooming into the AGM, when we looked at kind of the factors that we knew had to be in the HSCs, RUNX1, HOXA9, MLT3, and its target MECOM and HLF, we really were able to narrow down this one cluster that has to represent those nascent HSCs. And from here, we were able to pull other uniquely expressed programs and identify this kind of a nascent HSC identity, which we display in this scorecard that really shows how we can distinguish it from these other cell types in the AGM. And we were also able to narrow down that only this 6 gene signature was enough to identify the HSCs compared to progenitors at all stages of development. So from here, we could find that not only the early AGM region had HSCs, but also the placenta and yolk sac had HSCs before they have seeded the liver or are robustly in circulation. So when do the HSCs go to liver? Liver is the site where they expand and mature, which is very important for their developmental path. So using the same HSC signatures, we could now see that the transition between AGM and liver happens between 5 and 6 weeks development. So are they really the same cells? When we looked at all the different transcription factors that we associate with HSCs, we can see that they are already there at the AGM stage. So transcriptionally, they already know that they're going to be able to be the big shot, the HSC, but they don't know how to do their job. When we looked at the UMAT, we can see that there's a clearly temporal progression between the HSCs at different stages. So we wanted to look a little bit in more detail. What are these programs that evolved during HSC maturation? So there were some obvious ones. We could see that these endothelial and kind of shared megakaryocytic programs in the nascent HSCs in the AGM, they get downregulated as HSCC to the fetal liver. There's some inflammatory programs that get downregulated. There's a few uniquely known as fetal genes, such as IGFBP2, HMGA2, Lin28B, that also get downregulated at different stages of this maturation, as well as proliferation settles down, actually already in the fetal liver, and then in cord blood, they are actually predominantly quiescent. If we look at the programs that are upregulated, what really stands out is this MHC class 1 and 2 molecules, and in particular, the MHC class 2 molecules are turned on exactly at the times when HSCs become robustly transplantable in the fetal liver. And there's actually studies now showing that this is actually functionally linked to their reconstitution ability. We see this MLT3 and the MLT3 transcriptional program is strongly there. And then we see a factor that you may have seen in your own studies, PROM1 CD133, kind of thought as a stem cell marker in many systems. Interestingly, that also gets turned on exactly at the time when HSCs become transplantable. And because these are surface markers, we can now go into our fetal livers at different stages, zoom into the kind of what we knew as the HSC population, and now we can see how the expression of both PROM1 and HLA DRA class 2 molecules increase during developmental progression. So now we have both kind of markers that we can use to map where exactly are the HSCs in this maturation path, as well as understanding of some of these key programs that change, which are not so much in the transcription factor identity, but more in what these cells are doing and how they're responding to the signaling. So where do these extraembryonic tissues go? When we place them in the UMAP, actually they kind of go in between the AGM and the liver. And when we use our HSC maturation scorecard, we can see that these extraembryonic tissues are indeed in the middle. And when we use a module score from some of these key factors that we associate with maturation, again, they are between the AGM and liver. And when this maturation score switches from immature to maturity score at 8 weeks of fetal liver development, that's exactly when they become transplantable. So now we have much better ways of tracking when our HSCs have reached the functional maturity. So what about the origin? When we did UMAP analysis of what we knew was the HSC cluster, we saw that it actually made a connection to endothelial cells, and more specifically to the arterial endothelial cells, whereas the other hematopoietic cells represented here didn't make a physical cluster. And when we look at the HOXA code, which we knew that associated with the definitive HSC lineage, we can see that there's the same HOXA pattern in these nascent HSCs and then the endothelial cells in the AGM, whereas the progenitors are not HOXA pattern, exactly how we had predicted, even with the much more limited information. But this also gave us an opportunity to zoom into this transitional population that had features of endothelial cells expressing TDH5. It's turning on the transcription factor RUNX1 that turns on the hemogenic program, but it doesn't yet express the pan-hematopoietic marker CD45. So when we place this hemogenic endothelium between these other populations, we can clearly see that it's an arterial population. It has the same arterial markers as the other arterial endothelial cells. And there are some specific arterial markers that are very highly shared with the hemogenic endothelium and this population that we call the pre-hemogenic endothelium that is preceding the hemogenic endothelium. Oops. And there are only a few key differences that I'm highlighting here, such as the IL-33, which is downregulated in the hemogenic endothelium transition, and then upregulation of KCNK17 that is turned on exactly at the same time as RUNX1 in this hemogenic transition. So now we have kind of like a prospect of putative markers for these transitions. We were very lucky, our collaborators in Germany were able to find an actual intra-aortic hematopoietic cluster from the AGM, and now we could place our markers here, and we could see that IL-33, the pre-AG marker, is here in the endothelium surrounding the aorta. It goes down in these hemogenic clusters, whereas KCNK17 becomes upregulated exactly at this time. So, and then SPINK2 we use as a marker as the cell that is already transitioned to an HGC. So now reviewer says, it's really great. Can you show a few more replicas? And I said, no, I'm probably never going to see this cluster again in the section. So we thought, okay, let's give it a shot and let's try to do some spatial transcriptomics where we have maybe a chance if we capture this intra-aortic hematopoietic cluster, maybe we can look into it. So we got a 5-week embryo and worked with the TPCL and TCGB, and we were able to find a few sections where there looked like that there was a kind of a visible intra-aortic hematopoietic cluster, and these are the ones that are presented here in red. So if we look at our pre-AG markers, they localize exactly in the aorta and some of the other large vessels. LDH1A1, a key factor of retinoic acid signaling, which we had learned from our other studies. It's also there in these aortas. And importantly, KCNK17 and SPINK2, which now really highlight the hemogenic transition, we only see them in those sections that have these visible intra-aortic hematopoietic clusters. And when we zoom in, you can see this in more detail, KCNK17 expression as well as SPINK2 expression. So now we have this resource that we can go back and look at any markers at any time when we have more ideas what would be critical regulators of this process. So now I can say with confidence that when we look at these images, like, okay, yeah, that's an emerging human hematopoietic stem cell. And I think it's beautiful. So, but how do we really know that these populations are connecting? How do I know that this IL-33 positive cell becomes this cell? So this is where we thought we can use the multiome, which also incorporates the ATAC sequencing. So we can kind of do epigenetic lineage tracing between these populations. So now we are looking at IL-33, the pre-AG marker, and we know that it goes down between the pre-AG to AG transition. When we look at the nuclear RNA in these, we can see that there's still some RNA left also in the AG fraction, and when we look at the regulatory sites that are linked to its expression, we see them in pre-E, pre-AG, and AG. So this really kind of establishes the epigenetic relationship between these populations. So we have more confidence that these populations are giving rise to each other. And some other interesting example, RUNX1, which really marks the launching of the hematopoietic program. We can see the expression start in the hemogenic endothelium stage. But already in the pre-AG stage, we have some tickling in these active regulatory elements. But when we look at this repressive link here, shown in the red, we can see that the earlier populations have a repressive epigenetic mark in these in some of these regulatory elements. And exactly at this AG state, there's a handover from a repressive to an activating link. So it's kind of a tug of war between the positive and negative regulatory elements when this RUNX1 gene is turned on. And the same thing we see for PTPRC, the pan-hematopoietic marker. It's not expressed in the hemogenic endothelium, but you already see both an activating and repressive link to this PTPRC, kind of like just keeping the reins until it's ready to be launched. So we think we're just getting into this, but we think that this will give us a lot more insight to these relationships, what these cells are, where they're coming from, where are they thinking about going. And also we can look at upstream regulators when we look into more detail into these sites. So from this study, we've narrowed down the 6 gene signature that we can identify HSCs in all tissues and locations throughout human development. From this, we learned that HSCs are not only early on in the AGM, but also in the placenta and yolk sac, which may represent the first stages of where the HSC maturation is starting. We've learned about the maturation programs, both intrinsic and what's on the surface, and we have markers how we can now track HSC maturation. We pinned down HSC origin to the arterial hemogenic endothelium, and we have unique markers that we can use to identify these endothelial cells. And we've also, not showing it here, but we have an idea of the key signaling pathways that need to be, that need to do a switch in this time when they are moving through this arterial endothelium and then doing endothelial to hematopoietic transition. So, of course, now we're very eager to use this information finally to our ultimate goal. Where are those pluripotent cell-derived HS species in the human developmental hierarchy? So we worked with our long-term collaborators, Andrew Elephant's team in Melbourne, and they sent us the cells. We worked together with them and tried to optimize the conditions to AGM-type hemogenic endothelium. So we said, okay, let's see what kind of cells these actually are if we take completely hands-off and we're not looking at your favorite marker and ignoring all the others. We use this actin program developed by Fei-Yang Ma in Matteo Pellegrini's lab, and which uses machine learning to kind of against our reference library. So when we look into here, we have both this adherent and suspension fraction. In the adherent fraction, we have lots of mapping to these different pre-AG populations. And then we have mapping to an AGM HSCs. We also have some mapping to these different progenitor types that are generated in the embryo as well. And when we zoom into the HLF positive cells, HLF is the most specific single marker to find the HSCs. You can really see that they represent AGM stage. Some of the floating ones on the yolk sac and placental stage, there's very little evidence of any kind of maturation to fetal liver stages. And when we put them on our maturation scars, where we can actually look at individual genes, we can see that these pluripotent cell-derived HS species, they represent really these early AGM placental stages in their maturity. And we know that at this point, in human AGM, there's one transplantable HSC in the human AGM, because most of them still don't know even where to go and how to do their job. So of course, now it's an interesting question. What are these cells? Can they transplant? This has really been the goal in the field, but there's not even a single publication to date that can show, real transplantable multilineage hematopoietic stem cells. So this is the protocol from our collaborators. You induce mesoderm with morphogens, you pattern the Hoxsey pattern, you get them through the hemogenic endothelial stage, and then you take the hematopoietic cells. They use this tomato transgene that you can isolate. You can see they're nicely expressing these key markers, CD90, CD34. They can cryopreserve them to pull enough cells and transplant, and then let's go. And I am so excited to report that they are now finally in the stage that they're actually, fairly consistent reconstitution. You can see here with this tomato signal. And if you look at between the, what kind of lineages they can make, there are several recipients that show multilineage reconstitution. They are shown here. When they are high engrafted, they typically are multilineage, whereas there's some that are more individual lineages engrafted. And when you compare these lineage distribution to cord blood, you can see that it's not totally awkward, that all the lineages are represented. And here you can see very nice FACS profiles that these really are bona fide human blood cells of different lineages. But if you look at this, it's still inconsistent. Not every mouse is transplanted, engrafted, and many of them are only myeloid cells or myelolymphoid cells. So it's clear, it kind of represents the picture at this time of the embryo, they have these nascent HSCs, don't know how to do their job, and there's this myelolymphoid progenitors that are generated that are the first ones to seed the fetal liver. So what are the opportunities to try to make this better? So, oh, something is missing here. I don't know why, but it's not showing here in the projector, but I just have to tell you probably by using other ways. So if I look at now these, we have another scorecard where we can distinguish are our cells of the HSC lineage, or do they represent these early progenitors in the yolk sac, which are highlighted by these markers. And we can clearly see, also verifying the acting predictions, that these pluripotent cell-derived cells are of the definitive lineage. And we've showed that they're in the maturation, they are in the AGM stage. If we zoom into these markers, these pre-AG markers that we've shown now are very highly specific to the definitive lineage. We see some expression of IL-33, but it's not in every cell and it's quite vague. There is actually no LDH1A1, so the retinoic acid signaling is defective in these ones. They are bypassing it by stimulating that with the retinoid, but this is something that we really need to pay attention to. Is like, why don't these in vitro derived cells, why can't they kicking this signal? And if we look at the other signaling pathways that are kind of doing a switch exactly in this hemogenic endothelial transition, such as expression of Wnt inhibitors, TGF-beta inhibitor, TGF-betas, we don't see them in these pluripotent cell-derived cells. So I think that there is some kind of either inefficiency or discrepancy in this pluripotent cell stage. How many of the cells are going to the HSC lineage versus further to the lymphomyeloid lineage? And then I think that there's a lot more that we can do now to try to perfect this process, that make sure that they really go through the right type of hemogenic endothelium, which really paves their future success. So the two areas we're going to be focusing is the maturation, as well as really getting this signaling, the timing of the signaling in the aorta right, when they are making the definitive HSC rather than these transient progenitors. So next, I would like to a little bit take a look into the programs that might be involved in this maturation. So we're turning into our good old friend, MLT3. We've already published that MLT3 is one of the key factors that we observe is downregulated in the cultured HSCs. When we restore MLT3 expression to physiological levels, we can see that we can now expand fully functional transplantable HSCs over 12-fold. And these are not leukemic, they don't have lineage bias. And why is it that? Because it's how MLT3 works. MLT3 binds to these transcription start site of these key HSC transcriptional regulators, and it works with them with multiple protein complexes, such as the superelongation complex to regulate transcription elongation, as well as the dot 1L complex to maintain active epigenetic marks. And so what it's doing, it's maintaining the activity of these HSC genes to maintain their stemness. And we've now verified by IP mass spec that indeed also in expanded human HSC, cord blood HSCs, the same interactions are true. And we've also identified some new factors, new interacting complexes such as NERD complex, NCOR, that may be also repressive complexes. And we think that this is why MLT3 is not locking into a fade, because it's switching with these different complexes based on physiological cues, and then can help balance what the HSCs need. By coincidence, we also found another level of regulation that we didn't anticipate. When we looked into the regulatory sites in MLT3 gene, we not only found the standard TSS, but it looked like there was a second TSS that had all the classical epigenetic marks of TSS. Also, looked like that there was an enhancer next to it that had all the enhancer epigenetic marks. And when we look at the ADEX sequencing of the most undifferentiated HSCs in the fetal liver, both of them are accessible. And as they start differentiating, they are become inaccessible. So, and when we look at the expression, how do we know which isoform is expressed? This matches to an annotated transcript of this short isoform, which is only the tail end of MLT3. It doesn't have the Yeats domain that binds to chromatin, but it has these protein interaction domains, and it has unique alternative exons that we can now also find in RNA sequencing. So these little peaks here only can come from the short isoform. So we can see that we can verify the expression of the short isoform also in fetal liver and cord blood HSCs. And interestingly, it's the very same region of MLT3 that is also involved in the MLT3 leukemias. So we know that this region has power. So if we look at the expression, both during culture and differentiation, the short isoform is going down like the long isoform, and it's even more strictly regulated by the long isoform. And if we look at the development in these early hemogenic tissues, we can see by single-cell RNA sequencing that MLT3 is there in the pre-AG, AG stage. We didn't know which isoform. We can see that there's a little bit of expression here, but it's only coming from the long isoform. The short isoform peaks only come a little bit later in the fetal liver stage. So interesting also from developmental perspective. So why do HSCs need two isoforms? We answered this question by doing isoform-specific knockout, either for the long form, short form, or both isoforms. And if we look at the long form knockdown, we can see immediately that they start kind of like a differentiating prematurely. There's this appearance of this very robust 38 population just in a few days after culture, and then they lose the undifferentiated CD34 38 population much more rapidly than the controls. If we look at the short isoform knockdown, interestingly, when we zoom into the most precise marker of the transplantable HSCs, EPCR, there's actually an initial expansion of this population. And when we looked at the isoform knockdown for both, we see initially this expansion from the short isoform, and then ultimately this loss of this undifferentiated population from the long isoform. So it's kind of a chimera of this both of these things. So it looked based on these phenotypic assays that they're doing kind of opposing things in culture. So what happens when we transplant them? Very interestingly, neither of these knockdowns of the long or short isoform or both isoforms can give robust engraftment. With short isoform, we occasionally see a little something. We're trying to figure out if this is a breakthrough from the knockdown or if this is a real phenotype, but we can clearly see that both of these isoforms are required for robust function. So now we're, of course, very intrigued. What do they do? So we did single-cell RNA sequencing of these knockdowns at 5 days of culture, and we isolated the HLF positive cells that contain still the HSC-like population. And we can see the immediate differentiation in the long form knockdown. And when we look into these programs that are regulated by this individual, either the long or short form knockdown, we could see that actually they are indeed regulating these programs into opposing directions. The long isoform is needed to maintain translation and important transcription factors, as well as suppress splicing, probably related to differentiation, as well as metabolic processes. Whereas the short form knockdown looks the opposite, and we have an exact opposite effect. So this tells us that they indeed are acting kind of in opposition to each other, and this also identifies this metabolism and mitochondrial biogenesis and metabolism as an important factor that is regulated by these isoforms. So what about the overexpression? We have shown before that the long overexpression expands. HS species we can see here in green. The controls, they kind of expand and then they poop down when they run out of their endogenous MLT3 expression. When we co-express the short isoform, they still maintain this trend, but it's less. And the short isoform alone is not doing much. And this is an old RNA-seq data from fetal liver, bulk RNA-seq data, but with these isoforms expressed individually. But we can see again that they regulate same processes, but in different directions. Bringing this idea that they might be acting in opposition to each other. One very interesting gene that we found to be most dramatically regulated by these isoforms was IGFBP2. When you express the long, it goes up. When you express the short, it goes down. And this is actually interesting because IGFBP2 is one of these fetal genes that we show that goes rapidly down when the HSCs are maturing. It's known as a fetal marker. It's in highly proliferative fetal tissues. It's also upregulated in many cancers, including leukemias, and it's involved in insulin receptor growth factor signaling pathways. The uncultured fetal liver HSCs have already downregulated IGFBP2. We put them in culture, they kind of reignite this fetal program. Let's be young again, otherwise we can't expand. When you overexpress the MLT3 short, long this is boosted, when you overexpress the short, it's actually suppressed. So this tells us that maybe this overexpression of the short might be suppressing some of these fetal properties. And when we look at the timing, when the short isoform is induced, we can see here in the fetal liver, at 6 weeks of development, it's exactly the same time when the IGFBP2 expression is going down. We're trying to now quantify this expression and more globally. We're just working to find the right program. This is from Salmon, and we can see that the long isoforms are represented in these oranges, the short isoforms in blue, but also when looking at this kind of computational programs, not just like, oh, I see a peak that I like, these early tissues have predominantly long isoform, no short, whereas the short isoform actually becomes a predominant isoform in the HSCs during development, later in development, suggesting that it's there to kind of temper this symmetric self-renewal, active expansion, and getting them to transition into a more homeostatic state. And so why does the long isoform expression work if you need both? So interestingly, if you overexpress the long, it's not only binding to its own TSS, but it's also binding to the TSS of the short isoform. And it's maintaining like a foot in the door, so that when we're expanding the cells, we can now also not only get expression of the long, but also we get a boost of the short isoform, maintain the expression so that we can maintain fully functional transplantable HSCs. So to summarize, we've learned that there's not only one, but two very important isoforms of MLT3, and that they both are important for HSC function. They are developmentally regulated, and the short isoform is coming later in development and promotes this transition of the highly proliferative fetal HSCs to a more mature HSCs that acquire a homeostatic state. And both of them are needed, and they are regulating these metabolic switches, and probably the HSCs being able to switch between different fates. Both of them are downregulated in cultured HSCs, but if you maintain, if you overexpress the long isoform, you're getting both for the same price, because at least you can maintain the short form TSS accessible. So we really think that this is a key mechanisms how HSCs can kind of do different jobs at different times as needed. And we think that having both is important. It's like a car. You're not buying a car that only has gas or brakes. You need both. Even to drive forward, you need to have a little bit of brakes so that you can do this very thoughtfully. So what about in leukemias? We're now very interested in these isoforms and their balances and imbalances in different leukemias. And very interestingly, at least we have some evidence that in multiple different leukemia types, there might be a long form overdrive, whereas the short form is not present. And same is true for example, Down syndrome. They have this fetal-like myeloproliferative disorder, and they don't have the brakes. They just have the gas. And you know that driving that kind of car is very dangerous. So how are we doing with the time? I feel like I probably consumed most of my time. I had still a short little story, but I don't know if we have any time left. I can't see the time anywhere here. Okay, I'll try to be, succinct. So, okay, so now I feels like, we have solved it all. It's all good. Now we know how to do this. Obviously, we haven't. Even when we express MLT3, not every cell that has the HSC phenotype is engraftable. So there's still a big opportunity to do this better. So we go back to the drawing board. Where else are we failing? So one factor called MICT1 goes very dramatically down in culture, and it's not rescued by MLT3. So, and this kind of downregulation of MICT1, it really goes sharply down with the transplantability. And so what about MICT1? We still didn't know much about it, but when we look into these various gene expression profiles, we can see that it's expressed in the endothelium and the nascent HSCs in the AGM. It's in fetal liver core, but all stages, it's in these undifferentiated HSCs goes down. And when we zoom into those kind of like a newly defined phenotypes of the most self-renewing cell, MICT is always in the most self-renewing cell. And when we pull out the MICT expressing cells from our HSC populations, they represent these other programs that really tell that these are maybe the ultimate most undifferentiated HSC. So what does MICT do? We knock down MICT in our lentiviral knockdown system, and they basically these cells just can't do anything. There's no expansion of the HSPC population in fetal liver or cord blood. And when we transplant them, even when they still have an immunophenotypic population at day 5, when we transplant them, they cannot function. So clearly it's critical for HSC function. So what is MICT doing? We did another single cell RNA sequencing study, and we're zooming into those HLF positive cells. These are really the representing the true HSCs or HSC-like cells. And we can see that the MICT knockdown cells, they are downregulating many very important programs that we associated with HSCs, whereas MICT overexpression is improving these. And interestingly, even if there's an upregulation of many of these genes that we associated with proliferation, these cells cannot really proliferate efficiently. When we track this proliferation at the single cell level, we can see that they are much more sluggish to go through the motions of proliferation. And we see there's multiple imbalances in key programs that HSCs need for proper function. So what is MICT? We thought it's a transcription factor when we started working in it, but even if it has nuclear localization signal, when we looked at the localization in the KG1 cells or HSCs, they're actually localized in endosomes. And what do they do there? We had no idea, so we did IP mass spec, and we saw that MICT is interacting with multiple vesicle trafficking components, signaling factors, G protein subunits. So it's clearly not a transcription factor, but it's doing something probably in the endosomes. So what do endosomes do? They do endocytosis. So now we look at the rate of endocytosis, and in the MICT knockdown cells, we can see that actually they have an increased endocytosis when you lose MICT. And what is endocytosis? Endocytosis is kind of like a filtering and interpreting the signals that are in the environment that they can properly be executed in the cell. And we can see how nicely, if you overexpress now MICT1 in the cultured HSCs that are kind of starting to run out MICT, they're actually suppressing their endocytosis. So what if you don't have MICT1? What happens to signaling? We did phosphomass spec in endothelial cells, and we can see that there is really a hyperactivation of multiple pathways, especially the signals that are in the culture. They get like, wow, it's just a screaming for these cells. And although we can't do IP mass spec, phosphomass spec with HSCs, we can see this imbalance and hyperactivation of these multiple pathways. And here we verified the PAKT overactivation. So what we've learned is that the MICT is another key regulator that is selectively expressed in human ECs and HSCs. It's downregulated in cultured HSCs very rapidly. It's absolutely required for human HSC expansion and engraftment, and it's required to maintain HSC quality and for them to able to proliferate and expand in culture. It's localized in the membrane fraction of endosomes, and it interacts with vesicle trafficking and signaling components from multiple different pathways. And it's preventing the excessive endocytosis and signaling hyperactivation. So if we now look into what happens to endocytosis in culture, that hasn't really been looked at. So if you look at even within the most highly HSC phenotypic population, there's a huge upregulation of endocytosis, whereas the progenitors don't show this. So clearly the HSCs have to regulate endocytosis, and we can suppress this excessive endocytosis by maintaining adequate expression of MICT1. So what's really happening? So you put some growth factors, and then you say, oh, why don't we put a little more, just make sure that we have. What these cells are actually, they're just, everybody's screaming, flit, kit, at the end of the day, they're so overwhelmed, they just collapse, they shut down, they can't proliferate. So it's very easy to understand why this is not a fully functional hematopoietic stem cell. So this has illustrated another key factor that we need to pay attention to know that when we're expanding HSCs, that we don't lose it, for them to be able to respond to the niche and do their job. And now our focus is looking into how MICT can be regulated. So together with MICT and MLT3 isoforms, we have, again, novel markers to look for and new exciting avenues to to look for what more we can learn about HSC self-renewal. And I would like to say that almost everything that we have learned about human HSCs, we've learned because we failed. You have to try to fail. And even if this prospect of making a pluripotent stem cell from iPS cells seemed like it's a kind of an old phrase and nobody is any more excited about it, for me, it was an interesting intellectual opportunity to learn about human HSC self-renewal. But I'm actually getting pretty optimistic and confident that these things are going to come to fruition and we can actually use them one day. So many people to thank, wonderful lab members. Vincenzo Calvanese has started most of these studies on human HSCs. Now Anastasia Bevelina is continuing the short isoform study. Julia Aguade Gorgorio, the MICTI study. A very important former lab member, Sandra Caballero Garcia, Iman Fares, and then collaboration with Fei Ma for the single-cell RNA-seq study, and many wonderful collaborators here at UCLA. You can't do anything alone. It really matters to having a right team who are excited about these challenges. And I see here a nice spot open. If anybody else is interested, please contact me. So I will stop there and take any questions if there's still time.
Thank you, Hanna. That was outstanding. So many new things. And certainly if anyone in the room wants to ask a question, and if anyone on Zoom wants to put something in the Q&A, then I will answer that. But
So I've been past the queue.
great. Okay. Oh, and you're first.
Just before, it's really ridiculous. It's a microphone you can throw around the room. So, but it feels ridiculous holding it. Hannah, that's fantastic. That's 17 years worth of work in 51 minutes. I think that's really impressive. This long and short isoform of the critical factor strikes me that understanding what its particular splicing control is would be useful.
Yeah.
Is there a known set of splicing factors? And does your single cell sequencing tell you that some are deficient or expanded in the time frame in which this splicing needs to happen?
So one thing to note that there's actually a second TSS. There's probably some little fiddling with the splicing also, especially also like how long this untranslated 5' is. I think both are involved, but right now we think that it's the main regulation of like these big shifts come from the using these alternative TSS. And we can see, that's why we love to do also the multiome, because we can actually see that the short form regulatory elements are coming already in the hemogenic endothelium kind of. I already know this is where I'm going to have a big job one day, but they don't get turned on then. But one interesting factor that kind of keeps coming from multiple areas, that retinoic acid signaling is what we've linked to this maintaining this short form element. So I think that that's probably going to be, it's key in this early stage to kind of like at least get those elements open. It's probably not the full story, but we hope to really be able to look at these elements and the situations where they do get suppressed. We just made a very cool discovery, like we look at this myeloproliferative disorder in Down syndrome, and they seem to have a long form overdrive. So I think that will be like an in vivo experiment to look at how these are regulated in an abnormal fashion.
Excellent. So there's a question online, and then it overlapped with one of the questions I was interested in. So it seemed like the EB culture from the pluripotent cells maybe didn't quite capture the metabolic phenotype that you have from your progenitor cells. And so do you know about the role of metabolism in, like, is there an opportunity to restore that and see if it improves their function?
So I think metabolism it's like it's so big, and I'm kind of always tried to stay out of it. But now when this long and short isoform, it seems like the metabolic switches are some of these key things, and those are of course things that can be very quickly changed, and how HSCs are responding. So I think that the metabolism is absolutely critical. We didn't find like any kind of like if I just look at the HSCs that are formed in the pluripotent cell cultures, there's a progenitor next to it in the same culture that have a higher metabolism than the HSC itself. So even in the same environment, they have some intrinsic ability to regulate that. So I think that part of it is like, making sure that we culture them in the right way. But there's also something unique how the cells in the same culture condition, different cells regulated in a different way. So I think that there's a lot more to zoom into that. The signaling, I think, is the absolutely key, but which then of course translates to metabolism. But exactly all the details, I think that more work needs to be done.
Great. A question here?
Yes, I'm just curious, from the reprogramming field, you know, some iPSC are, even though they can form teratoma, they may not form healthy chimeras. So there's huge variation in terms of the quality of the iPSC. So do you work with like just natural embryonic stem cell, and do you see any difference?
So great question, because I think that, you know, the differentiation protocol is only as good as, you know, their starting cell was. So we work mainly still with the classical cell lines from like H1 cell line from our lab, but these studies were actually done with an iPS cell line that they had done in Melbourne, and they very well characterized and make sure that there's no weirdness in them. But I think that, yeah, it always remains the question, like, you know, that they don't change in culture and have acquire any of these abnormalities, because then it doesn't matter how well you differentiate. If they have intrinsic issues, then it won't work. But
You may have answered this already, but you mentioned environment. Are you trying co-culture with different tissues as a source of external interest?
yeah, so you know, these people have tried to make you know, stromal lines, and we tried them too, and they've kind of, you know, people publish them. Nothing really works, like, you know, nothing has come like a game break. Okay, there are some the best stromal lines still. I shouldn't say that's not true. The best stromal lines for our human cells were still mouse OP9, which everybody has in there. So we couldn't find anything better. But right now, I think that there's an opportunity. Now we can do Visiome. We can look at, we have like a 5 different subset of stroma in the AG, and we're starting to look exactly which stroma subsets are around the aorta versus more. So now we're starting to get there. And that's what we want to do with all these tissues. How is the stroma different in the fetal liver? Where is it really? What kind of endothelial cell is it next to? So I think this is, again, an interesting time to revisit the same idea with a lot more information what to go after.
Yeah, it's interesting you mentioned the MHC genes, and a lot of people are thinking about that in cancers in terms of boosting immunotherapy. Is there anything that we know about their regulation in that context?
Oh my God, I'm the wrong person to answer because I don't know anything about immunology, but yeah. So, but one thing that we are kind of looking at also, you know, just start to look a little bit at the regulatory elements, because it's an interesting way for, you know, I'm taking the nascent HEC and then the mature HECs, and I can see that this gene is getting turned on. So we can see that some of the positive elements are already on, but not others, and then there's a negative element. So again, you know, like, it would be a wonderful project for somebody to really delve into using the multiome and following with that. But right now I don't have much new to tell you.
Great question here.
So it's a very wonderful talk. Thank you for that. I have a really quick question. I noticed that you mentioned when you overexpress the MLT3, you got to achieve a 16-fold expansion. So
or around 12-fold in that study.
Okay.
But yeah.
So have you looked at zoom in the metabolic homeostasis on those 12-fold expanded HSCs to see if MLT have done anything?
So, okay, so when we looked at the cells like in the mouse, so they behave like the normal cord blood HSC. So they don't look weird, they don't proliferate more, they don't do anything. But, you know, if we take the culture that went in, you know, we put still a lot more cells than what engraft. So not every one of those cells that went in, it was the engraftable cells. So I think that now that we're starting to have more markers, MICT1 is one of them, and others that we can start, like, you know, putting the 500 cells that we put them in there and putting them into subsets, and then really try to see, okay, who were the ones who actually engrafted? And then maybe we can more tell prospectively, like, how are these different than the ones that don't engraft, even if they're within the same population that we think are the HSCs. But right now, I think that those are exactly the nuances that we still need to kind of understand to fully maximize the process.
Yeah, I guess my question is more following up than the microenvironment. because if it's achieve a more expansion, definitely their metabolism has been either speed up or
Yeah.
reconfigured. So I just wonder like how metabolism related to that 12-fold increase of the cell. Yeah.
Yeah. So anything that we put in culture, like they have to get activated. And I think that that's where this reactivation of this fetal program with IGF-PP2, it's probably kind of helping them. That it's kind of like, okay, if you're asking me to, run and dance and I have to kind of put that mode on, and then you're not in the sleeping mode. And so they refresh these fetal programs. When we look at the full scorecards, they don't become fully immature, but they kind of borrow back these programs. So I think that, and that all relates to high proliferation. And again, metabolism, everything is metabolism at the end. So I don't know specifically what question, but it's also in culture, short isoform gets downregulated faster. And so there is, I think some of them are, what is the chicken and the egg? But then if you go beyond that, you've turned off your short isoform, the regulatory elements are silenced. I think you can't anymore go back. The beauty when you are still in the HSC state, you can do the dance, you can sleep, you can dance, and you can still keep doing both. But there's a point, when they go too far, where you push them too far, and then they can't anymore go back and engraft and wait, sit and wait what they need if you want them to transplant.
All right, so 1 minute for the final question. How are the regulatory regions defined in the multi-ohm data?
Okay, so this is it actually, when I look at the loop file and I click show me the active and repressive links to this, it's the program is trying to tell me that, and it's telling that these links are linked to expression of this gene in either positive, like this peak is linked to the expression of these genes either positively or negatively. So it could either be an enhancer for that gene, or it could be a TSS for another gene that is then regulating it. So there's kind of like, it gives you some hints, but then it's up to you to kind of do the hard work and figure out what they are really, those connections. But I have to say, it's absolutely mind-blowing. Like it's addictive. When you start looking at it's following and like, you don't want to do anything else. I promise you.
Hanna, that was exceptional. Thank you so much.
Thank you.